So now we're going to go through all of the steps of meiosis. And meiosis, just like mitosis, has replication of chromosomes. So in interphase, we're going to go through the S phase, which is going to copy the DNA into multiple copies. Unlike mitosis, meiosis is going to have two cell divisions. So this makes sense. We're going to have two copies of DNA, but we need to cut the number of DNA in half. So we're going to cut it in half once with one division called Meiosis One, and then we're going to cut it in half again with the second division called Meiosis Two. So instead of ending up with two daughter cells that are exact clones like mitosis, we're going to do two divisions to end up with four daughter cells that have only half as many chromosomes. So we're going to start with one diploid cell, and we're going to end up with four haploid daughter cells, which will be our sperm or egg. So let's go through Meiosis One first. So Meiosis One is going to have prophase, metaphase, anaphase, and telophase 1, followed by cytokinesis to separate them into two cells. We're not worried about pro-metaphase at this point. You can take prophase and pro-metaphase and kind of collapse them just into prophase. So we've got P, M, A, T, prophase, metaphase, anaphase, and telophase. So up first is Prophase One, and this is going to, so Meiosis One is all about separating homologous chromosomes. So in the very beginning of meiosis, we have to find the sister chromosomes that belong to the same number of chromosomes. So if this is chromosome 5, this is chromosome 5. This is our homologous chromosomes. Those are going to find each other. So you can see here in prophase, we've got some other things that are very similar to prophase. We've got the centrosomes kind of migrating to the different poles. We've got the spindle fibers forming to separate those. We've got the nuclear envelope and nucleolus disappearing. But notice all of the chromosomes. The chromosomes have started to condense, and they are formed in their homologous chromosomes. So when the homologous chromosomes find each other, this forms what is called a tetrad, because it's got 1, 2, 3, 4 sister chromatids in a tetrad. Tetra means 4. And then I want you to look at what these chromosomes are doing. They're not just sitting right next to each other like my picture. They're actually forming little X's. They're crossing over each other at these little regions called chiasmata. And this is in preparation for something called crossing over. So crossing over is only going to happen during Prophase One. You absolutely need to know that. That'll definitely be on the test. And what happens is the chromosome from mom and the chromosome from dad find each other. And the chromosomes are going to cross over. And then a little portion of those is cut off, and they actually switch pieces. So the non-sister chromatids, those are the ones in the center next to each other. Non-sister means that we've got one from mom and one from dad. These are sister chromatids. Those are not exchanging because those are exact clones of each other. What we're doing here is we're creating some genetic diversity. We're going to switch chromosome pieces from mom and dad and make new pieces of chromosomes that are actually combinations of what we got from mom and dad. So this is called synapsis, which is where the DNA pieces actually break off, switch pieces with each other, and then reform. So if chromosome from mom is red and the chromosome from dad is blue, after crossing over, we're going to actually have new combinations of chromosomes where these two have switched places. And so now we have a sister chromatid that's purely from mom, a sister chromatid that's got mom and a piece of dad, a sister chromatid that's mostly dad with a piece of mom, and a sister chromatid that's completely from dad. So all four of these are going to create different versions of this chromosome in the ultimate daughter cells. So crossing over happens in tetrads in Prophase One. Next we have Metaphase One. So what's going to happen in Metaphase One is that the homologous chromosomes are going to line up at the metaphase plate. Notice that we are still in those tetrads. The homologous chromosomes are still associated with each other. Anaphase Two, the homologous chromosomes are going to separate. So at this point, we're going to separate. We're going to pull one full chromosome this way and one full chromosome this way. And notice again that you can see these little pieces where they went through crossing over, where this chromosome and this chromosome have slight differences now because they shared some DNA in crossing over. The reason that we have to do this first process where we separate homologous chromosomes is so that the daughter cells are going to have one copy of chromosome 1, one copy of chromosome 2, and one copy of chromosome 3. This one has one copy of chromosome 1, one copy of chromosome 2, one copy of chromosome 3. So this is going to ensure that the two cells that we make here have not just 3 chromosomes, but the 3 correct chromosomes. So in telophase 2, we see very similar. The homologous chromosomes are now in a new... The homologous chromosomes have been separated, and they're now going to form new nuclei. Sometimes the nuclei fully reform here and then have to break down again. Sometimes they don't completely fully reform because the cell is going to divide again. It just depends on the type of cell. And then eventually we'll go through cytokinesis and cleavage furrow, which will give us 2 daughter cells. These cells are haploid at this point. So the cell in Prophase 1 had 1, 2, 3, 4, 5, 6 chromosomes. So this has 2 pairs of n equals 3 chromosomes. So this is 2n equals 6. Each of these cells is going to only have 1 pair of chromosomes. So this has got 1, 2, 3 chromosomes, which is n equals 3. So by the end of Meiosis One, those cells are haploid. However, the DNA is still duplicated. So now we're going to need to divide again to break apart the duplicated DNA. Meiosis 2 is going to be very similar to Meiosis 2 is going to be very similar to mitosis. So in Meiosis 1, we had the separation of homologous chromosomes. Meiosis 2, we're going to have the separation of sister chromatids, which is just like what we did in mitosis. So again, the phases are named the same way, P, M, A, T. So prophase 2, and this is happening twice because we already divided once. The chromosomes are condensing again. The nuclear envelope is breaking down. The spindle fibers are forming and moving to the opposite poles. This way we're going west and east this time. And this is happening in both cells at the same time. Metaphase 2, the sister chromatids, are going to line up in the center. This should look just like it did in mitosis. So these cells start out with 3 chromosomes. These are haploid. This one is haploid, right? 3 chromosomes. Break apart here for a second. We have 6 chromosomes inside of a cell. So those cohesins that hold the centromeres together get broken, and the chromosomes are going to be pulled to the opposite sides. And then in telophase, each one of these cells is going to have 3 chromosomes. So meiosis 2, it starts haploid, it ends haploid. We actually go from diploid to haploid in meiosis 1. So again, here the nucleus is going to reform, our spindle fibers disappear, we're going to go through cytokinesis, and then we will end up with 1, 2, 3, 4 haploid daughter cells. Between our asexual reproduction and our sexual reproduction. So mitosis conserves the number of chromosomes. This is going to make clones. Whereas meiosis cuts the number of chromosomes in half. We go from 2n to n, haploid to diploid to haploid. Mitosis are going to have genetically identical to the parent. Again, these are clones. The goal of meiosis is to create genetically different individuals so that we can preserve variety in the population. In mitosis, if you've got a skin cell and you're just trying to repair skin cells, you definitely don't want to make different skin cells. You want to keep them exactly the same. Mitosis, we have the cohesins cleaved apart during metaphase. This means that we're separating sister chromatids. Meiosis, Anaphase One separates homologs, those tetrads, whereas Meiosis Two is going to separate sister chromatids. This is something that you absolutely should know. This will definitely be on the test as to whether you're separating homologous chromosomes, those pairs of 4, or just sister chromatids. Mitosis has one division that's going to result in 2 cells, 2 daughter cells. Meiosis has 2 divisions that's going to result in 4 daughter cells.